Good evening. This is the biological molecule experiment of our group. A biomolecule, sometimes called a biological molecule, is any of the numerous chemicals produced by cells and living organisms. Biomolecules come in a vast range of forms and sizes, and they perform a number of functions. The four major types of biomolecules are carbohydrates, lipids, nucleic acid, and proteins. So let's start. First, the ingredients that are going to be used in today's experiment are fresh lemon juice, which is going to represent our enzyme. Next, butter for our fats, and an apple for carbohydrates, yogurt, which will be our protein, and lastly, 3 plastic cups of our labeling and putting the experimental material that will be needed. The first thing we're going to do is squeeze the lemon until the juice comes out and put it into 3 plastic cups, making sure that the lemon juice will be the same amount as the other cups. Then, we will start by putting 1 apple cube cut into the lemon juice as well as butter and 1 spoon of yogurt. This will be labeled as fat, carbohydrate, and protein. After putting all the ingredients together, we wait for an hour to see what will happen.
1 hour later.
And the 1 hour stops now. We will check the before and after results of the experiment. So, as you can see, nothing actually happened in this picture. But we wait until the night ends and the yogurt, which is labeled as protein, has a lighter color than the other cups, which have made small progress. But other than that, nothing actually changed in a small period of time.
Hypothesis. If a slice of apple is placed in the first cup for an hour and there is no reaction or change as well as the second, which is butter, but the yogurt changes a bit of color for a whole night's observation, then the hour of waiting has nothing but a hint of change for all three of them that are classified as fats, carbs, and protein. Conclusion. It is concluded that the rate of chemical reaction increases as the substrate concentration increases. Enzymes, which is the lemon juice, can greatly speed up the rate of reaction. However, enzymes become saturated when a substrate concentration was high. It brings the reactant into contact with one another so that less energy is needed for the reaction. They are unchanged during the reaction so that they can work again. The materials that are being used are fresh lemon juice, 3 plastic cups, butter, apple, yogurt.
This is Miss Palmo, Mr. Landozabal, and Mr. Hilapad, members of this experimental vlog group, saying goodbye and thanks for watching.